Gene electrotransfer is a physical method used to deliver genes into the cells by application of short and intense electric pulses. In successful gene electrotransfer, several steps are involved. Formation of a complex between DNA and cell membrane. Translocation of DNA across the permeabilized membrane. Transfer of DNA from cytoplasm into the nucleus and gene expression. For gene electrotransfer, different electric pulse protocols are used in order to achieve maximum gene transfection. One of them is changing the electric field orientation during the pulse delivery. In this video, we demonstrate the difference in gene electrotransfer efficiency when all pulses are delivered in the same direction, and when pulses are delivered by changing electric field orientation. Experiments were performed on Chinese hamster ovary cells, CHOK1. For the experiment, follow these steps. Grow cells in a culture flask in the incubator for 24 hours. On the day of the experiment, prepare cell suspension by trypsinization with 0.25% trypsin EDTA solution and count the cells. Centrifuge cells for 5 minutes at 1000 rotations per minute at 4 degrees Celsius and resuspend cell pellets in isoosmolar sodium phosphate buffer to a cell density of 5 million cells per milliliter and move the sample to the appropriate dish. Cells are exposed to electric field in pipette tip with integrated electrodes connected to a high-voltage prototype generator. These electrodes allow application of a relatively homogeneous electric field in different directions. The tip and the generator were developed at the Laboratory of Biocybernetics, Faculty of Electrical Engineering, University of Ljubljana. Add plasmid EGF-PN1 to a cell suspension in concentration of 10 micrograms per milliliter. Mix cells with plasmid DNA thoroughly and incubate the mixture for 2 to 3 minutes at room temperature before applying electric pulses. Aspire 100 microliters of cell suspension into the pipette tip with integrated electrodes. In this experiment, a train of 8 rectangular pulses is applied using high-voltage prototype generator. 2 different electric field protocols are used. In the first protocol, all pulses are delivered in the same direction, whereas in the second protocol, pulses are delivered by changing electric field orientation. The second protocol can only be used with appropriate pulse generator, which allows application of electric pulses in different directions. Immediately after the pulse application, transfer the cells from pipette tip into 6-well plate and add fetal bovine serum, 25% of sample volume. Incubate cells for 5 minutes at 37 degrees Celsius to allow cell membrane resealing. Add 2 ml of HAMF12 to each sample in 6-well and incubate cells for 24 hours at 37 degrees Celsius in a humidified 5% CO2 atmosphere in the incubator. Efficiency of gene electrotransfer is determined as the percentage of cells expressing GFP 24 hours after pulse application. The cells are observed using a fluorescence microscope. In the acquisition software, set the excitation wavelength to 488 nanometers and choose an appropriate bandpass filter for green fluorescent image to detect the cells that are expressing GFP. Acquire at least 5 images, phase contrast and green fluorescence at 20 times objective magnification. Count cells in phase contrast image and cells that are expressing GFP in green fluorescence image. Determine the percentage of gene electrotransfer efficiency by dividing the number of cells that are expressing GFP with the number of all cells in each corresponding image. The percentage of cells expressing GFP when all pulses are delivered in the same direction and when pulses are delivered by changing electric field orientation is presented. By changing the electric field orientation, the percentage of cells expressing GFP increases significantly.